Hi there, I'm George Hammond, Senior Conference Producer at Technology Networks, and I'm here to introduce our next talk, Building the Next Generation of Cell Therapies. I'm incredibly pleased to welcome Dr. Andrew Mancini. Dr. Mancini is a cell engineering scientist and corporate development professional with a background in both technical research and business strategy in the cell therapy space. In his current role, Dr. Mancini leads both scientific partnerships and M&A initiatives as part of MaxCyte's corporate development team. He works with academic and commercial cell therapy developers to assess and structure strategic collaborations, leveraging his technical expertise in cell engineering and gene editing to drive deal making. A very warm welcome to you, Dr. Mancini. Following the presentation, we will also have a Q&A session, and we welcome any questions that you might have. Please ask questions using the Q&A system to the right-hand side of the video player. And so, without further ado, I would now like to hand over to Dr. Mancini to begin his presentation.
Excellent. Thank you, George, and thank you, everybody, for tuning in today. As George mentioned, my name is Andrew. I currently work at MaxCyte on the corporate development team, although I'm a scientist by training. And prior to my current role at MaxCyte, I was actually a field scientist working with cell therapy developers all around the world, helping them build the, I'd say, current generation of ex vivo cell therapies. Now, what I want to focus on for the next 40 minutes or so is really on how we can build the next generation of cell therapies. And when I talk about the next generation of cell therapies, what I'm really referring to is engineering the next 10 years of ex vivo cell therapies in particular. So this talk's really going to probably be a bit different than the other talks today, and is going to take more of a global view on where we are currently at in the ex vivo cell therapy space and how I think it's going to evolve in the next 10 years. And it's really going to be focused on these engineered cell therapies. So think about your CAR T cells, your TCR T cells, engineered hematopoietic stem cells. This is what I really want to zoom in on and look at where we currently are and where we're going. And so where I want to start is really current day, where are we when it comes to ex vivo engineered cell therapies? But before even jumping to 2025, I want to wind back the clock a bit to 2017. 2017, I think, is when we would start to say cell therapies became more mainstream. And in 2017, if you're developing an ex vivo cell therapy, there's a good chance that the process you are using to manufacture that cell therapy looked something like the following. You started with a patient with a hematological malignancy, potentially multiple myeloma. From that patient, you isolated CD3 positive T cells, which you then transduced with a CAR lentivirus so you could stably express your CAR sequence, probably in 2017, CD19 or BCMA CAR. These CAR T cells you then expanded over 7 to 10 days to create your final autologous CAR T product, which was then infused back into the patient. And so I say that in 2017, this is probably the type of process that you were working on if you were a developer back then, because in 2017, over 80% of engineered cell therapies were CAR T cell products. Over 90% of engineered cell therapies used a single lentiviral transduction step to engineer the cells. And over 90% of these products were autologous in nature, meaning you isolated cells from the patient and then redosed back to that same patient. So while there were a few other cell engineering strategies that groups were taking to engineer their ex vivo cell therapy products, in 2017, the general strategy looked as such. But we know in the past 8 years, things have changed quite dramatically. And what I want to talk about is what some of these shifts were in this process, which now lead us to where we currently are at for engineered ex vivo cell therapies in 2025. Before I dive into the data, I just want to talk a bit about where this data is coming from. So these data is really looking at a global view of the ex vivo cell therapy market. So these are all engineered ex vivo cell therapies in development, both preclinical and clinical development from the US and Canada, Europe, and Asia Pacific. So while completely understand that this doesn't capture every single program that's out there, I think this gives a really nice overview as to what the ex vivo cell therapy landscape looks like in 2025. So the first shift I want to talk about is going to what types of cells we're engineering. So again, in 2017, over 80% of engineered cell therapy products started with a T cell. In 2025, T cells still make up the majority of all cell therapy starting material, with over 250 programs in development using T cells as the starting cell type. However, we have seen a diversification in the types of cells groups are using to create their products. NK cells and iPSCs are the number 2 and number 3 most common cell types that groups are starting with now. And we've even seen an expansion beyond these cell types to hematopoietic stem cells, mesenchymal stem cells, and other cells that groups are using for their starting material and then engineering into their final product. So while in 2017, over 80% of the cell starting material was T cells, in 2025, 45% of all engineered ex vivo cell therapy products in development do not use T cells as the starting material. So this is one big shift which has occurred in the past 8 years. And this is probably not very surprising to many of you in the audience because there's quite a bit of news about groups developing CAR-NK products or using iPSCs for cell therapy development. The second shift I want to talk about is maybe a little less evident. And this is the shift from viral to non-viral. And so again, 2017, over 90% of engineered cell therapy processes used a single lentiviral engineering step to go from the starting material to the final product. In 2025, this is what the breakdown looks like. 38% of engineered ex vivo cell therapy products in development use one or more non-viral steps. So while lentiviral transduction maintains the most popular engineering method, and truthfully, I don't think lentivirus is ever going to go away for ex vivo cell engineering, we do see that for every new viral-only product that enters development, there are nearly 2 new non-viral or hybrid products entering development at the same time, which means as the years progress, we will expect this viral portion of the pie chart to get smaller and smaller as more groups begin incorporating non-viral engineering strategies into their cell therapy product. And so when we're talking about non-viral engineering strategies or hybrid cell therapy products, which incorporate non-viral and viral engineering strategies, what are we actually referring to? So what are developers actually using for the non-viral engineering? When we zoom in on this 38% here, we see that by and large, groups are utilizing nuclease technologies in order to engineer their cell therapy products. CRISPR-Cas is, of course, the most popular system that most developers are using. There's also several other nuclease technologies which groups rely on, such as zinc fingers and talons, which have their place in the non-viral engineering space as well. And we also see a significant proportion of groups using transposon-transposase systems for the gene integration step. I'll talk a little bit more about these technologies later on, but I just wanted to give you a preview as to how groups have added in more complexity into the cell engineering step by incorporating some of these newer non-viral engineering tools into their workflow. And so what the incorporation of these non-viral engineering tools has enabled is a variety of new engineering strategies to create cell therapies. So again, back in 2017, the vast majority of cell therapies were autologous CAR T products. And these are products which took a T cell and stably integrated a CAR transgene. This pie chart here shows that in 2025, we're at close to a 50-50 split between allogeneic in light blue and autologous in dark blue programs. And there's a few interesting things which you can actually find when you dig into the data that this pie chart represents. First, if we look at this autologous stable bucket, this refers to programs where you are stably integrating a CAR transgene into the genome, or you're using something like CRISPR-Cas to create an edit in the genome. So these are, I would say, permanent changes to the cell's genome. And there's 3 strategies that groups rely on to create these autologous stable cell therapy products, which are lentiviral delivery, transposase systems, and CRISPR strategies. Now, what's interesting and maybe overlooked quite often in the cell therapy community is that there's actually some cell therapies which we can classify as autologous transient cell therapies. So now, rather than actually making a lasting change to the cell's genome, we are just going to transiently modify the cell. In some cases, this is going to be transiently expressing a CAR sequence using mRNA. In other cases, this might be knocking down a gene temporarily by transfecting in an siRNA or an ASO. Now, this transient autologous bucket is quite a small minority of all of the cell therapies in development. However, what we do see is that year over year, there are more of these transient autologous cell therapies entering into development than the year before. And so we are starting to potentially see a shift towards groups who are trying to develop cell therapies without necessarily altering the genome of the cells. And I anticipate that by 2030, we're going to see this bucket represent probably close to a quarter of all autologous cell therapies in development. Now, on the other half of the chart here, looking at the allogeneic side, I don't think it's very surprising that 1 out of 3 allogeneic cell therapy programs are iPSC derived. Despite the challenges that the iPSC cell therapy space has run into in the past few years, the technology still holds a tremendous amount of promise, and I believe represents the future of cell therapy. I think what is even more surprising, though, is going back to the fact that nearly 50% of all cell therapy programs in development are allogeneic in nature. And again, back in 2017, it was about 90% autologous. Now in 2025, in 8 years, we've seen quite a strong shift towards allogeneic strategies, most likely reflecting some of the issues that we all know that autologous programs can face, including some of the manufacturing scale-out challenges, as well as some of the COGS issues associated with manufacturing autologous therapies. I believe that this allogeneic bucket is just going to keep growing as the years progress, and especially as we have more allogeneic programs progress through clinical trials and into commercialization. So already from 2017 to 2025, we've added on a couple layers of complexity onto the ex vivo cell engineering for cell therapies. We've talked a bit about there's different cell types groups are engineering. There's now non-viral engineering strategies groups are taking. they're using CRISPR nucleases, they're using transposons, and now they can use different cell materials and go after auto or allo strategies. There's one more layer that I want to add on, which is really further expanding the complexity that we are seeing for ex vivo cell therapies. If you recall, back on the first slide I showed, I mentioned that 90% of cell therapies in 2017 utilized a single step in the engineering process. In 2025, 50% of ex vivo engineered cell therapy products in development utilize 2 or more steps to go from the starting cell material to the final cell product. And shockingly, almost a quarter of ex vivo cell therapy products have 3 or more steps. Recently, we've actually begun to see ex vivo products that incorporate up to 10 distinct engineering steps in going from the starting material to the final product. And it's not just the number of steps, it's also how you order them and how you go about them, with about 50% of products preferring a serial engineering approach, where you do edit A and then edit B in sequence. And then the other half, they tend to prefer more of a simultaneous engineering approach, where you do edit A and edit B together. So that's one more added layer of complexity. And then the cherry on top is that most of these multi-step engineering processes actually combine multiple types of edit. So you might have a cell therapy product which is engineered with 5 separate steps, and 3 steps could be a CRISPR knockout, 1 step could be a transient mRNA expression, and 1 step could be a transposon integration. And so altogether, the reason I threw all of these data at you is to show you that from 2017 to 2025, the floodgates have really opened in terms of the complexity for ex vivo cell engineering for creating cell therapies. And so now, when you go and you look at an example cell therapy manufacturing process, it doesn't look like it did in 2017. Now we are starting with patients who also have solid tumors, patients with autoimmune diseases, neurological disorders, a wide variety of indications. And from these patients, it's not just T cells, it's NK cells, macrophages, and other cell types that developers are using as their starting material and engineering using a variety of different engineering tools, including both viral approaches and non-viral approaches, either by themselves or in combination with each other, to produce a wide range of different engineered cell therapies, which can be expanded and created into a final product that might be autologous, that might be allogeneic, or it might be even something more complex, a multi-cell product, or a product incorporating multiple types of CARs, or a variety of non-integrating and integrating approaches, all which then goes back into the patient. So where are we? In 2025, we are in a world where ex vivo cell therapy has become exponentially more complex, even compared to 8 years ago. And now that kind of begs the question as to, why in 8 years did it become so complex? And so what I want to talk about now is taking a brief look at how we actually got to this level of complexity, because I think this is really going to inform the future. Because if we look at the past 8 years and see as how we went from a relatively simple process into 100 different, very complex processes, we might be able to see what the future looks like. And so I am going to take the stance that the foundation of this complexity goes down to 8 gold standard tools that groups use for cell engineering. Not all of you may agree with this stance, and that's okay. I'll talk a little bit about some other enablers of complexity. But in my experience working with developers over the past years, the reason that I believe complexity in cell therapy has exploded recently is because of these 8 tools that I'm going to talk about. And these tools fall into 2 main buckets. If you think about what you need to engineer a cell, well, you need a delivery tool and you need an engineering tool. Delivery tools, we have our old reliable lentivirus. Again, you can quote me, this is never going to go out of style. I think it's always going to be one of the most popular delivery tools for ex vivo cell therapy. But it's been joined by some, I guess you could say, newcomers with AAV, which is predominantly used ex vivo in CRISPR knock-in workflows, as well as lipid nanoparticles, which are becoming more commonplace for gene editing, and what we consider the workhorse of non-viral engineering, which would be electroporation. On the engineering side, of course, with lentivirus, we have viral DNA integration as one of the most common engineering tools. In the past 5 years especially, we've seen an explosion in utilization of CRISPR-Cas editing in cell therapy products. And now CRISPR-Cas is almost synonymous with ex vivo cell engineering at this point. We have also, of course, seen transposase systems become commonplace, especially with groups who are not looking to integrate their gene of interest using a viral approach. And then, as I already mentioned, some groups are also employing mRNA to transiently express genes of interest as part of their engineering strategy. And so this is the toolbox that I would consider would be the gold standard tools that many groups are using to build their cell therapy programs. And I will argue that if we combine these 4 delivery tools with these 4 engineering tools, we can build almost any cell therapy process that exists in 2025, just with these 8 tools. To give you an example of this, I've graphed out here 3 workflows. Workflow 1 is going to focus on T cells. Workflow 2 is going to focus on NK cells. And workflow 3 is going to focus on iPSCs. And these are going to give you examples as to how you can mix and match these different tools to create a wide variety of final products. Our first and simplest workflow is what I would consider a quite common allo CAR T workflow. You start with your T cells. Your first step is going to be knock out 2 genes, which will give you some sort of immune cloaking. In this case, I chose TRAC and B2M. This would be done, for example, using electroporation to deliver CRISPR RNPs, one targeting the TRAC locus, one targeting the B2M locus. After the knockout, say 72 hours, you can then chase with a lentiviral construct to integrate your CD19 CAR. And this will, after 7 to 10 days of expansion following lentiviral delivery, you end up with your allo CAR T process, allo CAR T cells. And so this process by 2025 standards is actually not that complex, but compared to 2017, this is still an order of magnitude more complex compared to the standard auto CAR processes we saw back then. Now let's add a little bit more complexity with workflow 2. And we're going to do this by making it a fully non-viral workflow. So now instead of doing a integration using a lentivirus, we're going to take a common strategy that developers use for NK cells, which is delivering our transgene, which is a CD123 CAR, in the form of a transposon, and we're going to co-deliver it with a transposase. If you deliver these 2 components using electroporation, you will get stable integration of your CD123 CAR into your NK cells. And then as the cells are recovering and expanding, we can actually pulse in an LNP targeting NKG2A. So this LNP would have our Cas9 mRNA plus the guide RNA targeting NKG2A. And so in this process, which is fully non-viral, we end up with an NKG2A knockout CD123 CAR NK cell product, which is allogeneic. Again, not the most complex workflow, but I think it's a really good example as to how these technologies can be mixed and matched to create a non-viral workflow. But if we want to talk really about complexity, I think the king of complexity would have to be the iPSC workflows. And an example of this is, say you have your iPSC starting material. We're going to start again with a non-viral knock-in. In this case, we're going to use CRISPR to knock in a BCMA CAR into a safe harbor locus like AAVS1. So we'll deliver the CAR DNA alongside the CRISPR RNP using electroporation. And then we are going to do something which more and more groups are now doing, which are several rounds of gene knockout. And these are sequential gene knockouts, in this case using LNPs, where we have 4 targets we want to knock out, and we want to do one at a time so that we can avoid potential on-target translocations. This type of sequential editing strategy is used primarily to improve the safety profile of a multi-edited product. And so in this case, we will do the knock-in using electroporation, and then one at a time, pulse in LNPs to knock out each of these targets until we end up with our 6 times engineered iPSC cell bank, which can then be used for downstream differentiation, probably in this case, into a CAR T product. So these are 3 examples of workflows created from mixing and matching these 8 different gold standard tools. And one thing that I wanted to bring up when it comes to these tools, such as AAV or electroporation or LNPs, is that it's not that in the past 8 years that there were massive technological advancements that now made these viable strategies for ex vivo cell engineering. A lot of the reason that I think that these tools have become more commonplace is actually just that they're more accessible. And so there have been, of course, some fine tuning of the different tools, but I think where we are in 2025 is that it's very easy to get started with gene knockout using a CRISPR RNP compared to, say, in 2017, where the options were a little less accessible. And so, again, I don't necessarily believe it's the increase in complexity is due to massive technological advancements in terms of the technology itself, but it's more so due to the increase in accessibility of these technologies. And now, when we're talking about other enablers of complex cell engineering, obviously I am a bit biased towards the engineering step, but I do need to shout out that there are other advancements such as new cell culture platforms and new cell handling platforms, which have enabled us to culture at large scales cell types other than T cells better than ever before. There's also been improvements in the molecular tools that we use for engineering, such as guide RNAs and CARs, which enable us to take on more complex editing strategies, and also new analytical platforms so that we can better characterize our cells than we could even a few years ago. And importantly, and something which I think cannot be discounted, is that in 2025, there's actually quite a good knowledge base for ex vivo cell therapy. In 2017, truthfully, most of the groups, I believe, were more making it up as you go along. Quite a nascent field even 8 years ago. It's not that it's fully matured in 2025, not by a long stretch, but in 2025, over these past 8 years, there's been a significant increase in the accumulated knowledge as to how to build an ex vivo cell engineering process so that you can get a good product at the end of it. And this has enabled groups to go into their cell therapy development with more confidence, and with a clearer strategy than even a few years ago. And so, how did we get to the level of complexity that we see in 2025? And again, I believe it's key advances in the delivery and cell engineering technology, which has enabled many, many viable ex vivo cell engineering strategies. And again, not to sound like a broken record, but the advances in this case, I believe, are not just technical, but they're also in terms of accessibility of the tools. So the last piece that I want to talk about is now the future. We talked about 2017, we talked about 2025. So let's fast forward to 2035. Where are we going in the engineered ex vivo cell therapy space? And I'm going to say one thing which is not very controversial, and then one thing which I think is going to be quite controversial. We're going to start easy with the non-controversial piece. And so I believe that ex vivo cell therapy is going to stay highly complex. Because when we think about going from a starting cell material into an engineered cell therapy, there are a lot of things which need to happen before the engineering step, during the engineering process, and afterwards. At this point, with our developed toolbox of our 8 gold standard tools and all the knowledge that we possess in the ex vivo cell therapy space, I find it hard to imagine that we are going to significantly regress towards more simple processes. I don't think that's super controversial, but please let me know in the chat if you completely disagree. What I think is more controversial is that I actually think that 2025, this is going to be the least complex that things will be in the next 10 years. Because I think we are still at the beginning of cell therapy complexity. And I say this because what we are seeing in the field is rapid advancements in delivery technologies and engineering tools, which are going to enable more workflows for ex vivo cell engineering than are currently viable. And for delivery technologies, this includes a variety of physical delivery technologies, strategies like VLPs and exosomes, and even newer technologies like cell-penetrating peptides, which each have their own place in the cell engineering process. And then if you combine these with emerging engineering tools, including base editors, epigenetic editors, self-amplifying RNAs, and even newer editing systems like the newly described Tigger-Tass system, I believe we're going to be at a place in 10 years where the toolbox is not just these 8 tools that I show here. It's going to be closer to 20 or 30 tools that cell therapy developers can pull from and mix and match to create their ideal cell therapy product. And so if we're heading towards a place where complexity is only going to increase, and this in 2025 is the least complex it's going to be in the next 10 years, what's this actually mean for developers? So the way that I look at it is that cell therapy complexity is a double-edged sword. And so there are many opportunities associated with increased complexity for ex vivo cell therapies. If you have more flexibility and more options into how you can engineer your cells, you can build better cell therapies. You can create therapies with better efficacy, with more durable responses, and potentially with better safety profiles. However, as you increase complexity, there's also risks which become more and more common. Many of these risks are on the manufacturing side. With more complexity comes typically more risk of manufacturing failures and also higher manufacturing costs. Additionally, we tend to focus on the good things which can come out of engineering cells multiple times. However, we also must keep it in perspective that there are potential safety risks every time you go in and engineer a cell. And so it's going to be of utmost importance that we find ways to accurately characterize engineered cells, especially as we continue to move in the direction of more complexity. And so we want to access everything in this left column while minimizing everything in the right column. So how do we embrace complexity and get all the benefits of complexity while still managing risk during cell therapy development? And so I'm going to answer this question in a slightly roundabout way. And what I want you to think about it from is these risks really come in the form of roadblocks. And when we have more complexity in an ex vivo cell therapy engineering process, we are having more potential roadblocks. And these roadblocks really fall into two primary buckets. We have our technical performance roadblocks, which are things like low efficiency of cell engineering or poor scalability from research scale to manufacturing scale. And then we have our technology access roadblocks, which are more roadblocks in the sense of, finding ways to access the technologies that you need to create your cell therapy product. Each of these roadblocks have associated consequences if you don't deal with them. For technical performance, this tends to manifest in terms of lower quality final products and longer development times. For technology access, this most likely manifests in terms of higher costs of manufacturing, especially with royalty stacking, and also potentially being locked into suboptimal technology choices. For each of these roadblocks, we can actually put them on a very simplified version of the cell therapy development process and map them out to where they most often occur for the first time during cell therapy development. And then also map out the consequences, which can occur shortly after or further down the road. And so now, if we're thinking about increasing complexity, and increasing complexity can have benefits, but also lead to roadblocks, the question is, what can we actually do to avoid these roadblocks? And in the last couple minutes here, what I want to talk about is a couple strategies that groups take to circumvent these roadblocks so that they can add more complexity to their cell therapies while de-risking their therapeutic development. And the roadblocks I want to focus on right here are the early ones. These are all technical roadblocks, which tend to occur in the R&D stage or early on in process development. And these roadblocks typically come up because of choices that are made at the R&D stage in terms of the technologies which are being used to engineer the cell product. And these roadblocks come up. Groups may not necessarily address them at the earlier stages and then push it down to later. And then eventually they become a significant issue, most often during late stage process development, where now the whole process needs to be reevaluated and choices made at the R&D stage need to be rethought later on in development. This is not ideal because this tends to lead to significant delays in development as well as significant costs. And we're talking about 6 to 12 months of delays, as well as millions of dollars of associated costs. And so there are groups, though, who find ways to get around these early roadblocks. And they do so usually by taking two strategies. And these are strategies that, through my time working with cell therapy developers, we've seen many, many developers employ to de-risk these early technical roadblocks that can occur as they're adding more complexity into their process. Strategy one, I'm calling start with the finish. And strategy two, I'm calling leave no stone unturned. And so Start with the finish is something which I think most of you in the audience are probably familiar with. This is referring to looking at where you want to be at the very end and then making the decisions that you to get to that right now. In terms of cell therapy development, this means look at what we need in our final GMP manufacturing process and what we need to create our final cell therapy product, which is going into patients, and then start making the correct decisions from the earliest possible point in the R&D stage. So what I'm referring to here is that there are platforms which groups use and tools that groups use for their cell therapy engineering process. These are gene editing systems. These are cell culture systems, cell transfection systems. The earlier you can lock in your manufacturing solution during development, the less chance you're going to come across a roadblock at a later stage. Because what we have typically seen is that groups will start with an entry-level platform. Once they start getting towards process development, they'll switch to a PD platform. They start thinking about going into clinic, they'll switch to an initial manufacturing platform. And then when they need to scale out or scale up, they'll switch to their final manufacturing platform. Each of these switches can be risky and also can incur significant costs, and more often than not, lead to developmental delays. What we have seen instead that groups who are successful choose to do is they still start with the entry-level platform. But even as early as when they are wrapping up their lead ID and going into process development, they are switching over to what their final manufacturing platform will be. This allows them to avoid these costly platform changes and start using their final manufacturing solution at a much earlier stage. Some of you are going to say, I'm crazy, and that's why I want to add this small caveat / sanity check. Say you can't do this for every single platform, but the more that you can do it for, the better. And especially when it comes with more complex cell engineering, where you have not just your cell culture platform, but you might have 3 or 4 delivery methods and 3 or 4 engineering methods. The earlier you can lock in your final solution for these technologies, the better. This allows you to go through development with more confidence and de-risks these later stages of running into a roadblock. The second strategy, which I'm just going to briefly touch on since I'm running out of time here, is what I call leave no stone unturned. And what this refers to is spending a lot of time up front to really understand your process and your product to a level that even regulators don't need to understand. And what we have heard from developers, and these are just example quotes, are things like, if the FDA is not asking, I'm not going to check. Or, yeah, that's interesting, but we can look at that later. Or even there's things about my product that I actually prefer not to know. Because what some attitudes are in cell therapy development is that these are very complex products to begin with. And if you begin digging into it too much, you might find something which is going to impact a regulatory approval or impact just your developmental process and lead to delays. Now, what I would argue is that I completely understand that viewpoint, but what we've seen is that the most successful groups actually go above and beyond what regulators are asking. And they do things such as doing additional off-target testing for gene editing. They do things such as full functional annotations of viral and transposon insertion consequences. They look at genetic subpopulations and the impacts of genetic subpopulations on gene editing, and they go above and beyond on the characterization, relying on single cell analysis techniques so that they can really understand every single point of their product and their process. Because what we are seeing with especially more complex cell therapies is that the more you can learn about the product earlier, the less chance you're going to be surprised later. And there are groups that we know of that will do what is required, but then once you start expanding towards additional donors or really starting to go through higher throughput manufacturing, you can run into some significant barriers to the continued success of your programs. And so this idea of going above and beyond in the characterization as early as possible, I think is going to become more and more important as complexity keeps increasing. And so where are we going? I think we are going to a place where there's just going to be more complexity for ex vivo cell therapy, but there's also going to be more opportunities to make better therapies as long as we can manage this risk. And so I'm going to end there. And hopefully this was informative, even if it was maybe a bit of a different flavor compared to other talks. Just want to leave you with 4 simple take-home messages. First, ex vivo cell therapy has gotten much more complex in the past 8 years. Second, the technological advancements enabling complexity are not slowing down. Third, cell therapy complexity is here to stay. And finally, we believe that the cell therapy developers who can find ways to embrace this complexity while de-risking at the earliest stages of development will be the most successful. And that developers who can leverage this complexity will be able to produce some truly amazing cell therapy products over these next 10 years. So I'm going to stop here. I see there's a lot of questions on the right. I'm going to try to get to those now. If you have additional questions, feel free to connect with me on LinkedIn, or feel free to shoot me an email at my email address listed here. So thank you all for your time. And now I will go and handle, I think, a few of the questions.
Excellent. Thank you so very much, Andrew, for that fascinating talk. We really do appreciate you sharing your expertise with us today, and you are truly an expert in the field of cell therapy development. As with all the talks, we're likely going to have lots of questions, and as you mentioned, we have received some fantastic ones already, so I'll move straight onto the Q&A now. So Andrew, your first question, in the context where you were discussing the different levels of complexity, what is the typical quality control steps at each step to ensure gene knockout and knock-in?
So typically, if we're talking about an engineering step, a lot of groups will do something like Sanger sequencing or ramp-seq. So you get a genetic readout of the knockout and knock-in, and then also look at the cell surface marker for something which is expressed on the cell surface using something like flow cytometry, or occasionally using qPCR. And some groups are doing this at every single step in a sequential editing workflow. Other groups are doing this at the end of the entire workflow. I will say for iPSCs, typically groups will be doing this after each step, whereas typically for more immune cell-based processes, it tends to be go through the engineering process and then QC at the end.
That's a fantastic answer. Thank you so much, Andrew. Now, moving on to your next question, it's clear that this field is rapidly developing across the world, but in your opinion, which nations is the advancements in this area the most rapidly developing?
Oh, that's a really good question. It's so dependent on the exact technology. I will call out, let's say, for Japan, I think this is where we've actually been seeing a lot of the most exciting developments for iPSC-based cell therapy processes and really highly complex engineering of iPSCs. If we're talking about transient autologous processes, some of the work being done up in Norway and in Denmark is quite exciting there, as these groups have started to focus more and more on transient approaches to create novel cell therapies. Across the board, I'd say a lot of the innovation is still happening in the United States, especially in the Boston, Cambridge area, San Francisco Bay Area. But also now in China, we are seeing quite an explosion of companies using novel strategies to create cell therapies. So it's really across the globe, we are seeing different groups innovating. But there are specific geographies where specific technologies are moving faster than others.
That's excellent. Great. Thank you so much, Andrew. Unfortunately, we only have time for one final question today before we close out the session, which is adding to the mentioned shifts that you discussed in your presentation. We also saw a shift from fast-acting CD28 cars to more slow 41B cars. So there is an immense effort to improve the car persistence. How, in your opinion, do transient cars fit into this scope? And isn't it, I suppose, a step back?
Yeah, I think that's a great question, especially because I think there is a lot of skepticism around the transient car approaches, specifically due to this point. Um, in my opinion, and this also gets to potentially the question of, above, talking about transient therapies versus stable therapies. I think there's going to be situations where we find that a fast acting CAR is actually going to be potentially more beneficial in targeting the tumor, especially potentially when combined with other, co-expressed factors via mRNA. But then there's going to be other situations where the stable approach and having slower acting CARs is going to be more beneficial. I don't have all the answers on that, but that's a good question to think about. And, feel free to follow up with me after if you want to discuss more.
That's excellent. Thank you so much, Andrew. But unfortunately, that is all that we have time for today. Just a quick reminder to our audience that any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. So that just leaves me to say a massive thank you once again to Andrew and for addressing all of those fantastic questions from our audience. And to everyone in the audience, we'll be back soon with the final talk of the day. Thank you.